You've just taken your daily scoop of collagen, and somewhere in your mind, there's a picture. These molecules acting like tiny builders, traveling through your bloodstream straight to your wrinkles, your skin, your joints. But your stomach has other plans. To your digestive system, that expensive collagen powder is just protein, no different from chicken or eggs. So here's the question we need to answer. Does collagen actually reach your skin? Or is it simply broken down in your stomach and eventually excreted as very expensive waste? Spoiler, science says yes. Collagen does have an effect, but in the way marketing explains it. Collagen is a massive structural protein with a complex triple helix structure. Its molecular size is far too large to pass through the intestinal barrier intact. The moment you ingest it, digestion begins. Stomach acid denatures the protein structure, and proteolytic enzymes, primarily pepsin in the stomach and trypsin and chymotrypsin in the small intestine, break it down into smaller components. Ultimately, most of the collagen is reduced to amino acids, primarily glycine, proline, and hydroxyproline. At this point, the main criticism of collagen supplementation appears. If collagen is broken down into the same amino acids as any other protein source, then why pay more? Why not just consume meat, eggs, or other protein-rich foods? From a purely nutritional standpoint, this argument is logical. However, it assumes that digestion is always complete and that all protein is reduced exclusively to free amino acids. This assumption is not entirely correct. The key difference lies in the form of collagen, specifically hydrolyzed collagen. Hydrolyzed collagen is predigested through enzymatic processes into smaller peptide fragments. These include dipeptides and tripeptides that are structurally stable enough to survive further digestion. Among the most studied are proline hydroxyproline and glycine proline hydroxyproline. Research shows that these peptides can pass through the intestinal barrier and enter the bloodstream intact. This changes their role completely. Instead of acting only as building blocks, these peptides function as signaling molecules. Once in circulation, they interact with fibroblasts, the cells responsible for producing endogenous collagen. The presence of these peptides is interpreted by the body as a signal of collagen breakdown. In response, fibroblasts increase collagen synthesis. This mechanism is indirect. You are not delivering collagen to the skin as a structural material. You are activating the biological pathways responsible for producing it. The next question is whether these peptides actually reach target tissues, such as the skin. Studies using isotopically labeled collagen peptides have demonstrated that after ingestion, these peptides appear in the bloodstream and subsequently accumulate in the skin. They can remain in skin tissue for up to 14 days after consumption. This confirms that collagen-derived peptides are not only absorbed but also delivered to relevant tissues. However, this process is regulated by systemic priorities. The human body allocates resources based on survival needs, not cosmetic outcomes. If there is a demand for amino acids in essential systems, such as muscle repair, enzyme synthesis, or organ function, these processes will take precedence. Skin is considered a lower priority tissue. Therefore, the effectiveness of collagen supplementation depends on the overall nutritional status of the individual. In conditions of protein deficiency, the body will not prioritize collagen synthesis in the skin. Collagen supplementation can fail under several common conditions. The first is vitamin C deficiency. Vitamin C is an essential cofactor for the enzymes prolyl hydroxylase and lysyl hydroxylase, which stabilize and cross-link collagen fibers. Without sufficient vitamin C, collagen synthesis is impaired at a fundamental level. In practical terms, This means collagen intake without vitamin C significantly reduces its effectiveness. The second factor is dosage. Clinical studies indicate that the minimum effective dose for skin-related outcomes is approximately 2.5 grams per day, while joint and connective tissue support typically requires closer to 8 to 10 grams per day. Doses below these thresholds are unlikely to produce measurable effects. The third factor is the form and type of collagen. Hydrolyzed collagen peptides have superior bioavailability compared to non-hydrolyzed forms such as gelatin. In terms of types, collagen type 1 and 3 are most relevant for skin, while type 2 is associated with cartilage and joint health. Using the wrong type reduces specificity of effect. The fourth factor is glycation. High sugar intake leads to non-enzymatic glycation of collagen fibers, resulting in structural damage, reduced elasticity, and accelerated degradation. This process directly counteracts any attempt to support collagen through supplementation. The fifth factor is digestive efficiency. Adequate stomach acidity is required for proper protein denaturation and enzymatic breakdown. In conditions of low gastric acid, protein digestion is impaired, which reduces the formation and absorption of bioactive peptides. This directly impacts the effectiveness of collagen supplementation. From an analytical perspective, collagen is not a direct delivery system for structural skin repair. It does not function as a filler or a cosmetic substitute. However, it is not biologically irrelevant. Collagen supplementation provides a specific amino acid profile and, more importantly, bioactive peptides capable of modulating cellular activity involved in collagen synthesis. Its effectiveness depends on correct usage and physiological context. The most effective forms are hydrolyzed collagen peptides, derived from marine or bovine sources. Marine collagen is typically richer in type 1 collagen, which is more relevant for skin, while bovine collagen provides a combination of type 1 and 3. Recommended dosing ranges from 2.5 to 5 grams per day for skin-related outcomes, and up to 10 grams per day for joint support. Collagen should be combined with adequate vitamin C intake to support enzymatic processes involved in collagen formation. Equally important is proper digestive function, including sufficient stomach acidity, to ensure effective breakdown and absorption. Consistent use over a period of at least 8 to 12 weeks is required before measurable effects can be expected.